This video lecture is on the biological functions of select amino acids and peptides. Amino acids can be derivatized to make neurotransmitters. Neurotransmitters are substances that are involved in the transmission of nerve impulses. For example, tryptophan is a precursor of a neurotransmitter called serotonin. Serotonin has a sedative effect, which gives a pleasant feeling. Very low levels of serotonin are associated with depression, while extremely high levels produce a manic state. Some mental illnesses can be managed by controlling the level of serotonin and its further metabolites. Selective serotonin reuptake inhibitors treat depression by elevating serotonin levels in the body. Tyrosine and phenylalanine are precursors to neurotransmitters called norepinephrine and epinephrine, both of which have stimulatory functions. Epinephrine is commonly called the flight or fight hormone and causes the release of glucose and other nutrients into the blood and also stimulates brain function. Other biological functions of amino acids include glycine. Glycine is very water soluble and often is added to other molecules to make them more water soluble for urinary excretion. Many drugs are oxidized in the liver to compounds that contain a hydroxyl group, which is then conjugated to glycine. This final product is removed from the blood in the kidney. Arginine is part of the urea cycle, which is a metabolic cycle that removes nitrogen from the body. Methionine is used to make a coenzyme called S-adenosylmethionine, or SAM, which is a carbon donor in metabolic reactions. Glutamic acid is used to make monosodium glutamate, which is a flavor enhancer in many Asian and other foods. MSG causes a physiological reaction in some people that includes chills, headaches. Glutamic acid also acts as an excitatory neurotransmitter in the body. Insulin is a peptide hormone that is released from beta cells of the pancreas in response to high blood sugar. Insulin has many roles, including lowering blood sugar in the body by increasing sugar uptake from the blood into the cells, and regulates the metabolism of sugars, fats, and carbohydrates. In type 1 diabetes, the beta cells are destroyed by the autoimmune system, and so insulin cannot be released. Type 1 diabetic individuals have high blood sugar and must control it with insulin injections. Insulin is synthesized as a mature pre-proinsulin form. Modification of it, including cleavage and oxidation of cysteines, matures it into the final insulin peptide, which is then secreted from the beta cells. Oxytocin is a hormone that is associated with pregnancy. Binding of oxytocin to oxytocin receptors induces labor in pregnant women and controls the contraction of the uterine muscles. During pregnancy, the number of receptors for oxytocin in the uterine wall increases. At term, these numbers is great enough to cause contraction of the smooth muscle of the uterus in the presence of oxytocin that's produced by the body toward the end of pregnancy. When this happens, the fetus moves towards the cervix of the uterus because of the strength and frequency of the uterine contractions. The cervix stretches, sending nerve impulses to the hypothalamus. When the impulses reaches this part of the brain, a positive feedback loop leads to the release of more oxytocin from the posterior pituitary gland, and the presence of more oxytocin leads to stronger contractions of the uterus, such that the fetus is forced through the cervix and the baby is born. Oxytocin also plays a role in stimulating the flow of milk in a nursing mother. Glutathione is a tripeptide composed of glutamate, cysteine, and glycine amino acid residues. The peptide bond between glutamate and cysteine is unusual in that it is between the side chain carboxyl group of glutamate and the alpha amino group of cysteine. Glutathione acts as a scavenger for oxidizing reagents in that it reacts with them. It works that it itself becomes oxidized instead of other important biomolecules. And so 2 molecules of glutathione reacts to form an intermolecular disulfide bonds between the cysteines in the molecules, resulting in an oxidized glutathione molecule. Aspartame is the sweet peptide. It is composed of aspartic acid and phenylalanine residues and is of considerable commercial importance. A methyl ester derivative of this dipeptide is called aspartame and is marketed as a sugar substitute and is about 200 times sweeter than sugar. Both amino acids in the peptide have the L configuration. If D-amino acid is substituted for either amino acid or both, this results in a derivative that is bitter rather than sweet. Aspartame was approved by the U.S. Food and Drug Administration in 1981 after extensive testing. Diet soft drinks sweetened with aspartame carry warning labels of the presence of phenylalanine. This is very important for people who have phenylketonuria, also known as PKU, which is a genetic disease of phenylalanine metabolism. Phenylketonuria, or PKU, is an inborn error of metabolism since it involves defects in the DNA of the affected individual. In normal individual, the enzyme phenylalanine hydroxylase converts phenylalanine to tyrosine. However, people with PKU typically have defects in this enzyme such that they are unable to metabolize phenylalanine to tyrosine, so phenylalanine builds up and is eventually converted via an alternate pathway to toxic metabolites phenylpyruvate or phenolactate and phenylacetate. PKU can lead to abnormal development and mental retardation. If PKU is detected at birth, a modified diet can be followed, such as limiting phenylalanine and supplementing the diet with tyrosine, which can assist in normal development.